Welcome to our last session for lunch break. So I'm Catherine and I'm part of the Medicine Quality Research Group. And I'm going to talk a little bit about our project now. So 8 of us are based in Oxford. And most of us are actually pharmacists, but we also have doctors and data and computer scientists in our group. 3 equal students and also 3 honorary members. Our head of the group is Paul Newton and our deputy head is Celine. And we are very interested in the epidemiology of substandard and falsified medical products. We want to find out more about the impact they have on patients, but we're also interested in how they are related to antimicrobial resistance development. Another thing we're very interested in are innovative screening technologies to detect these products, but also new forensic tools that can tell us hopefully more about the origin and the sources and the trade routes of these products. So the project that deals with these forensic tools is called FORESFA. Then we have another one, the Vaccine Identity Evaluation project that uses or repurposes devices to screen substandard falsified vaccines. We're also working on a dashboard for portable screening tools called Decibel. And you've probably heard about the cases of contaminated paracetamol and cough syrups, mostly for children. So they were contaminated with the ethylene glycol and led to the death of many children. So we're also looking into screening tools that can detect these substances on a target product profile. We were involved in the Abacus project and did a medicine quality study in Ghana, Mozambique, Bangladesh, and Vietnam. And we're also involved in the Appian project. So that's an app that will facilitate medicine quality surveys. So ensuring the quality of medicines is still one of the urgent health challenges of our day of today. 1/3 of the population still has no access to essential medicines, vaccines, or other essential health products. And this vacuum is very often filled by substandard and falsified medicine. So that WHO estimates that 1 in 10 products in low middle income countries are actually substandard or falsified. And as you can imagine, that has a huge impact and lots of severe consequences, not only on the health of people, but also on a lot of socio-economic impacts and economic impacts. That's why the access to safe, effective, quality assured and affordable medicines is part of the United Nations Sustainable Development Goals. So I've talked a lot about substandard and falsified now already, but I haven't really explained what we mean with substandard and falsified. So important to know is that we focus on public health. So we are not interested in the protection of intellectual property rights. That's why WHO doesn't use the term counterfeit any longer. We have 3 separate classifications. So we have substandard, unregistered, and falsified medicines. And they are very different. So substandard medicine is an out of specification medicine that fails to meet quality standards. And that can be due to a mistake during the production, but it can also happen because it was improperly stored, or it can happen during transport or storage. But a falsified product, that's very different. So that's a medical product that deliberately or fraudulently misrepresents its identity, composition, or source. So there is a criminal intention behind this product. Very often, as you can imagine, that's difficult to differentiate between substandard and falsified, because how can you prove a criminal intention? So it's actually difficult, but still the origin and solutions are very different. So it makes sense to differentiate between the 2 of them. And one thing that can help us to classify a medicine as falsified is the visual inspection. You can see here a falsified product. It was easy to identify this product as falsified because it shows an outdated falsified WHO Essential Drug Program logo. But if you don't know that, it can also help if you speak French. So if you just have a close look at this label now for a few seconds and count the spelling errors you can detect. So there are quite a few. So that can help us to identify a falsified medicine. Here we have 2 coartem samples. One of them One is genuine, one is falsified. Who of you thinks that the left one is a falsified one? Just raise your arm. One? Yeah, a few more. Who thinks the right one might be a fake? Yeah, also one, two, three. It's the left one. So the packaging looks really identical, and only you can see the color of the tablet is a little bit different. It's a little bit of a lighter yellow. But these tablets, they did not contain any arsenite or lumefantrine at all. So these two products I just showed you are analyzed during the Forest Fire project. So in this project, we are working together with experts from very different fields. So from forensic genomics, chemistry, archaeology, social network analysis, and modeling. And our aim is to find out more about the origin and the trade routes of falsified antimicrobials. And we're using, therefore, forensic techniques, but also social network analysis. And we hope that the new forensic techniques we are developing can later on be used by regulators, enforcement, academia, or maybe also the pharmaceutical industry. And in the second part of the project, we're also interested in the role that substandard and falsified antimicrobials play in relationship to patient outcome and also the development and spread of antimicrobial resistance. That's that I mentioned before. We have team members all over the world, but most of us are based in Oxford. And only a few weeks ago, we had our third annual project meeting here in Oxford. So now I've said that we can use forensic tools to find out more about the origin of falsified medicines. But how can we actually do this? So the first thing we are doing is to just look at the packaging. So that's what we just did. So we do a visual inspection. We can use screening tools to find out more about if a specific project is actually similar or very different from another one. They might come from the same source or different sources. And then we can use many sophisticated methods such as DART, EDXRF, that will tell us more about the composition of a tablet. But we can also use isotope ratio mass spectrometry, and it will tell us more about where the water in a tablet actually comes from. And also the excipients such as maize starch or chalk, it can possibly tell us which region of the world these excipients come from. But then if you want to know more about where this medicine was actually manufactured, we can look at the environmental DNA we can find on the tablets, but maybe also behind the label of the packaging. And another thing we're doing is we look at the packaging itself. So for example, the plastic container where the medicines come in. So with that, we can find out if actually the packaging itself is falsified or if it's just a reused genuine one. And we also look at the dimensions and the structure of the tablets to find out more if they were made from the same tablet press or different tablet presses. So that will all give us some ideas about the history of these tablets. During the pandemic, we also started to look at substandard and falsified vaccines, not only COVID vaccines, but also all sorts of other vaccines. You can see here some of the compounds that were actually found in falsified vaccines. So amikacin, for example, sodium chloride or hyaluronic acid. And in the vaccine identity evaluation project, we are repurposing different technologies to detect substandard and falsified vaccines. So we are using, for example, MALDI-TOF that is widely used in hospitals to identify bacteria, but also SORS that you might have seen at airports. That's a non-destructive method to screen vaccines. I'm sure all of you have used lots of lateral flow assays, so I don't need to explain you what this is. And we're also using like very low-cost techniques such as urine stripe, ethanol stripes, or cholesterol matrix. And only a few weeks ago, there was paper published by the WE crew. You can see that here. So that was the first time that multi-MS and machine learning was used for vaccine authenticity screening. And you can see here at the bottom that they were successfully able to distinguish the hepatitis B vaccine from different liquids that were previously found in falsified vaccines. So as you have now seen, there are lots of challenges, and how are we gonna tackle them? So the WHO uses the prevention, detection, and response strategy, and if we now focusing on the detection part, and look what is possible. In the real world, we unfortunately don't have enough labs available that are well equipped. We don't have enough lab technicians to work in these labs. And of course, money and time are limited. That's why nowadays, or in the past years, screening technologies are becoming more and more popular. So with screening technologies, we are able to assess the quality of a medicine in a very fast, easy, and relatively cheap way. But it's important to keep in mind that they cannot replace compendial testing. So they can just help us to identify suspicious products that then have to be further analyzed. You can see here, there's a whole plethora of different screening devices available. And unfortunately, we don't have one magical device that can screen all products and is suitable for all settings. So that's why we are trying to help medicine regulatory authorities to decide or to find out which is actually the best device for which purpose. So, and we are doing this by updating the 2018 review on screening devices to find out more about what is available. And this data is then used to create a dashboard for portable screening technologies. So a dashboard, that's a digital interactive tool. And we are only adding portable screening tools, so they have to be portable, like one person is not to carry this device, and it has to be like an easy setup and to be granted. And this dashboard is called Daffodil, and you can see the prototype here. So that's still ongoing. And in spring this year, we had a workshop at Jesus College, and we had around 80 participants and lots of medicine regulatory authorities, also from low middle income country, and researchers that work in this area. Because we want to make sure that Daffodil, the dashboard, is actually useful to them, that it answers their question and helps them in their decision-making process. And a few weeks ago, we had an online advisory group meeting, and we are in the future also planning to further assess the utility and usability of this dashboard. And before I stop, just quickly wanted to mention, if you want to see some real falsified medicines here in Oxford, you can go to the History of Science Museums. They have a small exhibition on falsified medicines. And I also wanted to mention that we have our next informal medicine quality meeting in 3 weeks' time. So if you're interested to join this meeting, we will have around 40 participants, all from the area of medicine quality. Just talk to us during the coffee or lunch break, or drop us an email if you're interested to join. So thanks so much for